Welcome everybody to the Image Processing and Image Analysis workshop. My name is Sebastian Rief and I will be your host for this session. This workshop will be split up into 3 parts of each roughly 30 minutes. Between the parts, we will have breaks of 10 minutes in which you can ask questions. You can also ask questions in the chat during the sessions. Please ask them in the open chat for everyone so that everyone can profit from the answers given. The input data required to this workshop and also the result that we will be producing is pre-installed on the workstations in the cloud. So if at any point along the way you get lost due to some technical issues, there will be points where you can rejoin the process. Now a short introduction of myself. I studied mathematics and worked for 6 years in the Department of Mechanical Engineering at the University of Kaiserslautern before I joined Math the Market in 2018. Until 2021, I worked as an application engineer in the support team. A year ago, I moved to the development team where I am responsible for ElastoDict and the new BatteryDict degradation module. In this workshop, we will tackle the topic of importing images into GeoDict and processing them, and also running some analysis on the segmented images. The goal at the end is that you know how to import image data in GeoDict, how to apply image filters and segment the images. Finally, you learn how to analyze the imported data, for example, how to obtain grain size distributions and grain diameter distributions. We will use 2 different datasets in this session. The first one is an NMC cathode of a battery from the Battery Microstructure Library of the National Renewable Energy Laboratory of the USA. That scan was done by the University College London Chemical Engineering Department. The second one is an air filter media, such as you can purchase for a car. For this sample, we ordered the scan ourselves. Now let's get started with the workshop. If you haven't already done it, open workshop.cloud.mathtomarket.de in your web browser. Then use the credentials that you received via email to log in. The credentials are your email address, which you use to sign up for the workshops and the user meeting, and the password you receive via the email. Now let's log into the cloud interface for the workshops. If you haven't already done it, just log in with the credentials you have received via email. And then the cloud interface opens. It might take a bit longer for you since you log in the first time, maybe, but in principle it should work the same as here for me. Then here below you see already a list about the steps we have to do to log in here, and we will follow them now. At first we want to start a workstation here with the green Start Workstation button, and this workstation is started with the data which is contained in the zip file here, which contains all the data for all workshops. So you just have to click on Start Workstation. And then you see here that a workstation is starting here under the running virtual workstations. Then there is some additional information like the host name and the number of cores and the memory, but that's not too important for you. Below you see also that you can only start one workstation, so one should be enough for the workshops here. Then we can already copy the password for the workstation, so just click on the green show button here, and then you can just copy the password. One important additional information: after you finished working on the workstation, please terminate it with the red X button here at the right. This is the only way to shut down the computer. So please don't click it now because it shuts down the machine. But after you're finished with the workshop, please click this X button and terminate the machine. So now the status button turned to a connect button and is green. This means the workstation is ready and we can connect to it. So if you haven't already done it, go here under show password and copy it, just to make sure. And then click on the green connect button. And then the no VNC interface for the workstation is opened in another tab. And then you can click here on verbinden, that's a German word for connect or connect in your case. Paste the password. And then click on send password and then you're logged into the workstation. You see the desktop of your workstation in the cloud. This is like a normal PC running Linux. First, let's set the desktop to, or the computer to full screen. So we have the small arrow here at the left. Click on the arrow and then you can select the icon for full screen. Click on that and you get the workstation in full screen, and the resolution is also adapted. You can click again on the arrow to hide this again. Then everything is already ready and set up, so we can directly start with Geodict. Double click on the Geodict icon down here. Please do this only once. It takes several seconds until you see the Geodict splash screen. So now we're here in the Geodict main user interface. I just switch to full screen because it's a bit nicer. So what do you see here in the Geodict interface? So here is a large area which is later used for visualization. Then over here you have the current status of your computer, so the CPU load or the processor load, which is 0 at the moment, and here the usage of your main memory and of your hard disk. Below, in the project status section, you see, for example, your project folder. Here the default is Geodict My First Geodict Project, and then some information about Geodict, object, a bit structures, objects in your structure, result files and volume files. Currently we have nothing loaded, so it's all empty here. Below you see the currently active module, which is in this case FiberGeo. If it is contained in your license, FiberGeo is the default module at startup in Geodict. That means that it is automatically selected at startup. The first thing we will do is to set up our project folder. By default, it is always the folder My First Geodict Project in your Documents folder if you're under Windows, or in your Home folder if you're on Linux. But since all the data of your current Geodict session is saved into this project folder and you want to know where it is saved, it is always recommended to choose the project folder in the beginning. You can either do this by right-clicking on the project folder, click on Select Project Folder, or up here with the folder icon, click with the left mouse button on the icon and click on Select Project Folder. You can again see here the folder is selected as My First Geodict Project, and here we have everything already prepared. So there is already a project folder set up for you. For this, go inside here in the folder Workshop Materials 2023, open it and then select the folder Workshop or WS Image Processing and Analysis. And in there is a folder called Project Folder. Please select that one. So double click the folder, then you see it's selected up here. And perhaps I just go one folder up. There's also another folder called Results M2M. And this contains all the results that I created. So if you later want to compare results to my results, or if anything went wrong, you can find all the data in there. So select again the project folder. Make sure that it's selected up here, and then click on Choose. So now you can see that we have selected the correct project folder. Now everything is set up correctly, and we want to start by importing a structure. So by import images from a cathode, and for this we will use the import GeoVol module. Up here, click on Import GeoVol. Then if you selected it, so click on Import GeoVol, the module below here changes to Import GeoVol instead of FiberGeo before. Then we only need to click the Browse button to browse to the files that we want to load. So I click the icon. It opens by default in my project folder, which is nice here because the data is also contained in this project folder. And here in the folder called Cathode NMC 398NM is all the image data. So you can open it by double-clicking, and then you see that there is a lot of image files. It's several hundreds for all the slices of that structure. You can select one of them, it doesn't matter which, and then click on Open. Now the geometry import dialog opens and you can choose several options. For example, you can directly choose to crop the data, which we won't do here because we want to do it later in the image processing dialog. So here you could do this with crop X and crop Y and choose the size of your file here. Then it's important that the box import all files and folder is selected. Otherwise you could select a range of file names in there. But in this way we only need to select one file and the rest is selected automatically. Next, it's important that we select the correct voxel length because Geodict cannot know it from the image files alone. It's quite nice here that in this case the voxel length is written into the folder name. It's 398 nanometers. So for setting up the voxel length, we need to change the unit to nanometers. It's up here, the unit chooser. Select NM for nanometers and then type in the 398 for 398 nanometers. Now we're good to go and can click on image processing. This just will take some time until everything is loaded, and then the 3D image processing dialog will open. So here you see the image processing dialog. At first we will hide it because I want to show you something directly in Geodict. Because now you see something in the visualization area. At first it's only black, and that's because we're in the 2D view and we see the first slice in the X direction of our structure. And around the structure there's just some empty space, and this is shown in black. To look deeper into the structure, you can, for example, use the slider here for the slice, to slice through the structure. Now you see that there is a lot more interesting stuff going on inside of the structure. But you will directly recognize that the contrast is not very good. Everything is very dark here. Therefore, we will directly change the color map here, and we can do this up here by switching over to the volume field tab. So over the visualization area is a volume field tab. Select that, and then you can see here the color map settings. To change the settings of the current color map, which is called gray value, click on the Edit button here. Color map, gray value, and then the Edit button. And then you can see the histogram of the current file. And you can directly see the range here is very, very wide, but the interesting part is only going on in this small part here. Therefore, we can change the range. So you can drag these red boxes, take the one here and put it closer to the interesting part of the histogram, and we do the same up here. And you can directly see that everything got brighter and you see much more of the structure. To close the color map dialog, just click on close and the changes will persist here. For the 2D view here, you can now still scroll through the structure, and you see a lot more than before. But you could also select the directions which you see the structure, which was first selected for the SDX direction. You can change that to the Y direction, for example, or Z, if you want to see the structure from top. And that's it mostly for the 2D interface. And now let's switch to the 3D view. Up here, above the visualization area, you can see this icon for 2D, which is selected. Now we switch to 3D. So click on that button and you will switch to the 3D view. It will take a moment and then your volume field will be rendered in 3D. Here we are. First, you see the structure or the volume field directly from the top. You can rotate the structure or volume field by clicking and holding the left mouse button and dragging the mouse. So here you can rotate the structure. You can zoom either stepwise with the mouse wheel, so just scroll the mouse wheel, or continuously by holding and dragging with the right mouse button. So click and hold the right mouse button and then move the mouse, and then you can zoom in here. And additionally, you can drag the structure by clicking and holding the mouse wheel, the middle mouse button, and then move the mouse. If you want to see the inner part of the structure, maybe in this case because there's a lot of black around here, which is not very interesting, you can clip inside of the volume. Go to the volume field tab above here, and then at the right you have the clip sliders. You can hold and drag these sliders and slice inside of the structure. So as for example here in the Y direction, I can then see what's inside of the structure. You can do this in all the directions, so you could also crop it in Z and just to your liking. What might also be interesting to you is if you want to have a quick estimate of the thresholding on the structure, you can directly threshold the volume field in the visualization. You can see up here there's this area threshold by gray values. First I recommend to deselect the smooth button here. And then you can go over to the threshold slider and drag it. And then you can directly see at the first step all the black space disappears because the values are just 0. And then if you move the slider really carefully, you can then get an idea of the segmentation in the structure. So you can see that the grains begin to separate, and you get more of a feeling how that structure looks. But now we want to do it for real, so not just some preview in the visualization here. So we drag the slider back down to 0 and switch again to the image processing dialog, which you can find up here. Now it is in Windows in the taskbar below, but here click on the Geodict, the windows for Geodict, and select the image processing dialog. First I will maximize the dialog. It's with the rectangle up here. And now we're in the 3D image processing dialog. You can directly see 3 areas with the structure from the different views. The Z direction, so view from the Z direction, from the X direction, and from the Y direction. At the beginning you see the middle slice of all directions. One of the first things you will notice is that everything looks very red here, which is a bit weird. This is because by default you get an overlay shown here of the segmentation, and since there's a lot of black area around the structure, the automatic segmentation only segments the outer area from the inner area, and everything in there, or the whole rest of the structure is shown in red, so which marks that this is segmented as a structure. For now, we don't want to see this overlay, and therefore we change the visibility of the overlay up here. So you have a slider for the visibility and drag that completely to 0. If you want, there are also other overlays available, but for now we just keep the default of the segmentation. Then, if we see the structure now better, I can show you the main controls. You can zoom by holding the control key and scrolling the mouse wheel. So hold the control key and then you can stepwise zoom with the mouse wheel. With the mouse wheel alone, you can slice through the structure. So this works for all directions. And you can also zoom continuously by holding and dragging the right mouse button, as in the 3D view. So click and hold the right mouse button and zoom continuously. Additionally, you can also pan the structure with the sliders at the side here. Now let's get started with the image processing. We don't really need all the black space around the structure here. So the first thing we will do is to crop the structure. So for this we will go over here. And you could either choose image processing or segmentation and labeling. First, we want to start with image processing to make our image ready for the segmentation. So here in this tree, we click on the image size subtree, so on the small arrow in front of it, and then open the crop option. And in there, we could directly use the voxel numbers to crop the structure, or the slider. So if you move the slider, I can directly see that there is a yellow line moving, or a yellow rectangle, which shows which part of the structure is cropped out. You can also see that the overlay up here changes from segmentation to crop. We could also switch back to segmentation and the yellow line disappears. I switch back to crop because this is what we want to see now. And the yellow line indicates how the structure is cropped. And we can either change it with the sliders, as I have already shown, or you could also pick it up with the mouse and drag it. So now I zoom a bit in that we can see the structure completely, and now we choose the box around to find a nice part inside of the structure. So let's start here with the view in Z direction and select a box here somewhere inside. And do the same in the other directions. Sometimes the yellow lines are a bit hard to grab. And I think this looks already quite nice in the beginning. And we want also to make sure that everything is homogeneous in the structure. So I think this looks quite nice because we don't want to have these areas here, for example, in the view in the X directions. Now you can see by using the box here to crop, we change the numbers here. We will adapt them slightly just because it's just to have some numbers which are easier to remember, which you can then, if you want, copy and use in your case to reproduce my results. So we change the X direction to 250 to 750. And then the y direction from 200 to 700. And the z direction we go from 225, 225 to 485. This makes sure that we have all the same structure. And then we should get a structure of 500 by 500 by 260. And then to apply the crop, the only thing you have to do is to click on apply. So click apply. And now you can directly see that all the black space around the structure disappeared, and we only get this nice crop out of the structure. You might have already seen that the contrast in here is a lot better than it was initially in Geodict. This is because the gray value range is automatically adapted so that the areas at the borders are a bit cut out. Therefore it's a lot brighter, and you can also have a look at the histogram, which is more detailed than before because it's much more zoomed in. But in there you can still see that there is a large area where basically nothing happens from here 0 to around 2000, which is not really used, and we still want to take that out of our visualization. We can do this up here in the upper left corner by selecting the gray value range. And here instead of the 0 we type in the 2000 and hit enter. And it looks a bit nicer, so the contrast got even better. Now what you might have already seen is that everything is quite noisy in here, and therefore we want to filter our image to get rid of the noise. But before we do the filtering, we want to go into the first break. Before this, I will show you how to save the volume field. If you got lost during the process here, we can all start by loading the volume field in new and doing the filtering. For saving, go up here to File, and then you could either choose to save it as a 2D image stack or save a 3D image. A 2D image stack will create a lot of files and is usually larger and takes longer to load, so therefore we choose to save directly a 3D image. So click on Save as 3D image, then keep the default Geodict raw file .grw, which contains already some information from Geodict. So the header contains, for example, the voxel length, which makes the file then easier to load. Then select the compress option, which makes the structure smaller and faster to read and write. And then you still only need to click on Save 3D image, click on Save. And the remaining thing we have to do is choose a file name. We call it here cathode NMC cropped. We click on Save. And now the structure is saved and we're ready for the break. After the break we meet again to load the gray value image and filter it. I will remain in the chat here, so if you have questions just ask, and see you again in 10 minutes.